All right. Hello, everybody. I think it's just about 7 o'clock. Always trying to start on time. And I just realized as I was looking through the slideshow and was not able to make any changes anymore, that I made a grave, grave mistake by misspelling our speaker's last name. I apologize, Evie, for that mistake. I'm really sorry.
No worries. No worries.
All right, welcome to the Fleet Science Center's Suds Science talk. I'm Andrea. I have the pleasure of working for the Fleet Science Center. Some of you know me already. I'm excited about that. I'm gonna let some more people in. For everybody who doesn't know me, welcome to Suds Science. Suds Science is a monthly talk series with the Fleet Science Center. We normally host these on the second Monday of each month in the evening at a bar. Clearly that ain't happening anytime soon. So we moved to the virtual world in, I think at the beginning of April when all this madness started. But thank God this is possible. Otherwise we wouldn't even have this opportunity to get together. If this is your first Suds Science talk, Suds Science is meant to be more of a conversation than a lecture. So we will have a presentation by our speaker tonight, but we will have way more room at the end for conversation and questions. So I hope you will be enjoying some conversation and joining us for questions and discussion around the topic that we're going to learn about tonight. At the end of the talk, or closer towards the end, I will pull up a little poll for you guys. We always like to hear how you receive the events, how you like the events, and we need some demographic information. So I'm going to pull this up towards the end of the talk. If you could take a second to fill out that poll, that would be awesome. Everybody who comes to Suds Science regularly knows there's always a survey at the end because we love data. It is science after all. I will also let everybody know what's coming up in the upcoming months on science talks for the fleet at the end of the talk. Because now we would like to get to our speaker. We are very excited to be joined again by someone from the Sanford Burnham Previs Medical Discovery Institute. They've been a wonderful partner for us for Suds Science and our other series. And tonight we're joined by Dr. Evie Tan. Hi, Dr. Evie Tan.
Hi, can you hear me?
Yes.
Okay, so I'm Evie Tan. You can call me Evie. You don't have to call me doctor. So currently I'm doing research in the Hansen Lab at the Sanford Burnham Previs Medical Discovery Institute. So I'm a postdoc. So I completed my PhD, I think 3 years ago. So currently I just continue my career, hopefully to go into the academia field in the future. So, and before I begin, I would like to take this opportunity to thank the organizers, Andrea, and also Fleet Science Center for actually providing us this platform that enable us to share our knowledge with all of you. So today, what I'm going to discuss is that I am going to talk about a recycling process that takes place in our cells, and then show you how this process can be really important for our body to fight against aging and diseases. So hopefully at the end of our conversation, hopefully you can appreciate that in the cell, we have this recycling process that takes place that help us to take care of some of the trash that you don't even realize that it exists in the cells. So next slide. So we study aging, and at the lab, yes?
Sorry, I think you'll need to share your screen.
Oh,
you haven't shared it yet.
Oh, okay, I haven't shared yet. I thought I shared, sorry.
No worries.
Let me...
The joy of online talks. You know, there's so much to consider and do. It's really...
No, thanks for telling me.
Yeah, sure thing.
So this is my slide. Actually, this is my introduction slide. So, okay, so at the lab, let's talk about aging now. So at the lab, we work really hard to try to understand the process of aging. So aging is a fundamental process for all living beings. For example, I myself aging on a daily basis. So you can see, I hope you can appreciate, this is the younger version of me. And this is also me. It's still very young, but definitely older compared to this picture. And that's how I know that I'm going through aging. So while many of us scientists like myself are trying to figure out how to slow down aging, at least at this point, we know that aging is an inevitable process and not even the force can stop it. So why are we trying to slow down aging? Well, it is true that aging comes with a lot of good stuffs like wisdom, life experiences, perspectives, and should be celebrated. But aging do come with some of the stuffs that we don't want to. Like, in fact, aging is a major risk factor for many diseases like cancer, neurodegeneration, stroke, or heart disease. And the question that we have been trying to address is, why is aging a major factor for these diseases? And how are they relating to each other? Does neurodegeneration come first and then aging, or aging comes first and then neurodegeneration? So we are trying to ask why. And one way to tackle these questions for us is to first look into what happens at the cellular level and then find out what are the cellular processes that change during aging. And then hopefully we can follow these changes and then find out if we can tweak some of these processes and maybe to slow down aging. So here is a picture that I prepared to kind of explain the human physiology. So we have a picture of a human, which we already know that the human body is composed of organs. And in these organs, they are made out of a lot of human cells. So it's like a group of human cells and then comes together and then made up these organs. And then in these little human cells, they are composed of different kind of components, such as fat, cellular components like mitochondria. So mitochondria is like the battery in the cell that helps to produce energy to help keep the cells alive. And then we also have other proteins or enzymes. In this case, I will call them proteins. So like if you know of the stomach, you know that stomach can produce some sort of enzymes that can help to digest food. So you can think of these enzymes like proteins. And our cells change over time. And when these components can start to become damaged, and if not taken care of properly, these damaged components can be detrimental. And here is a picture that I show here. You can see that there's a sick and unhealthy cells because all those components are being damaged over time. And luckily in the cells, we have a recycling process called autophagy. So autophagy is important in recycling these damaged components. And however, the efficiency of this process also changes over time, just like the things that I talk about. So cells change over time, so as the autophagy process. And at the lab, this is a cellular process that we are interested in understanding and figuring out. And we are trying to figure out how to make it better so that it can help us fight against aging as well as age-related diseases. So a quick overview of what is this recycling process like. So the recycling process in the cell is actually very much like the recycling process that takes place in the environment. So which first, we are the consumers, right? We produce different kind of waste, like the organic waste, batteries, plastic, papers, glasses. And all this waste has to be recycled, else it will be detrimental for the environment, for us. But in order to ensure all these type of waste are being sorted, are being recycled properly, first it has to be sorted properly. And what helps to can help us to sort this waste is by the bins. We have different kind of color-coded bins that help us to better sort this trash, such as plastic trash will be in this particular bins. We can sort it in. Paper trash, we can sort it in this color-coded bins. And additionally, we will need the respective recycling trucks to come and pick up all these different kind of trash or waste, and then send it to the appropriate recycling factories for the recycling to take place. And this entire process and organization is very important, as if the trash are not being sorted properly, or if the trash are not being transported to the right place for recycling, it can impede the recycling process. And that is something that we don't want, right? So, and did you know that the recycling process in the cells has a lot of resemblance with the functions of the recycling in the environment? So here's why I say that. So in the cell, we also produce cellular trash, because the cells can go through different kind of changes, like maybe the food intake you take, your lifestyle, your different kind of habit, and the property of the cells can change, and sometimes the components of the cells can be damaged, and such as now the fat, if it's not taken care properly, it can turn into kind of toxic fat or bad fat, or like components that can be damaged, and then the proteins now are not being folded properly and they become sick. So we will need different kind of components that comes together and help to sort the trash and then bring the trash to the appropriate recycling factories. So we also have, in the cell, we also have components that acts like the bins and the recycling trucks in the environment, to take care of the cells, the environment of the cells, to make sure that they are clean and the cells are healthy. So what if the recycling process is not functioning properly, like I mentioned before, and I emphasized on the importance of how these organizations of the recycling process has to be taken care properly, because it's just like the example of a clean room. So you can look at this clean room like the recycling process is being organized properly. You have all the trash being sorted and then being taken out to recycle. And then when that process is being impeded and something is going on that's making that to become impossible to do so, then you have a messy room, and I mean, to us, sometimes we might still be able to tolerate the messiness to some levels in the room. However, in the cells, it's a different story. They're very fragile, and if the cells recycling process is not being taken properly, the cells cannot tolerate that, and they can become really sick. So you can appreciate that these cells, like when it is healthy, it's because like all the recycling process is taking place in the right order. But then like when that recycling process is being disrupted and no longer being able to function efficiently, then you can have a lot of cellular trash being accumulated, and then you can have cells that are looking really sick. So indeed, this is very true. Like many of the age-related diseases have a characteristic with very poor ability in recycling, in autophagy. So including cancer, heart attack, stroke, and neurodegeneration. So almost all of them are associated with the cells, because remember I mentioned that the organs are made up of a bunch of cells. So like most of these diseases are associated with the cells have accumulated with certain type of trash. For example, like heart attack has been associated with accumulated of a lot of bad fat in different areas of the organs that the fat is not supposed to be there. So like neurodegeneration has associated with a lot of accumulation of bad proteins. Proteins are not functioning properly, and they just accumulate in the brain organs, impeding the cell to be healthy and live finely So, and while many of us scientists, we realize this characteristic, and we have been trying to figure out how to find ways to boost the recycling process. Maybe by boosting the recycling process now, we can help to clear out this trash and make the cells become healthier. However, we all face a big challenge, which is that we don't know all of these beans and trucks yet in the cells. We know of this process, but we don't know what are these components, what are the name of these proteins yet. And so here comes the climax of my talk. So, which I'm going to disclose what my research is really about. So my research is basically to help identify these beans and trucks in the cells. Ta-da! So, and one of the approach that I'm trying to take is that here's what I believe. And not only I'm the only person who believes this, but I think in a lot of the aging community, especially in the autophagy field, we believe that by identifying these beans and trucks, a tick now. So let's imagine now I have identified one of these beans and one of that trucks, and then for sorting and transporting the specific type of waste associating with certain type of trash that has been known to kind of associate with, for example, like neurodegeneration diseases I mentioned earlier, has been associated with accumulation of bad proteins in the brain. So let's say I have now identified the trucks or the beans that can help sort or even transport the bad proteins to the recycling factories for recycling. Maybe we can now genetically introduce more of these specific trucks or beans to boost the specific types of recycling. And hopefully, this could be one of the approach that can allow us to help fight against or even slow down disease progression. So you can see now imagine that we have more of these trucks and beans to pick up the bad proteins to the factories for recycling. Now the cells will have less of the trash, and then maybe they can take some time to recuperate and slow down the disease progression. So, and in the lab, we have a really cool model organisms that not many general community, like people in the general community is aware of. Which is a tiny worm called the C. elegans. So this tiny worms is not the earthworms that you can find in the big long earthworm that it's like in the dirt that flip the dirt to make your dirt become more nutrients for the plants. This is a very tiny worms. They are microscopic worms. It's like 10 micrometers. So this is like one of you have to look at the worm under the microscope to see this worm. So we are using this worm in the lab to identify the beans and trucks that I just told you about. So how do we do that exactly? So these tiny worms are very powerful model to study aging, first of all, because they have very short lifespan, and they also have very similar recycling process like the human. And many aging scientists, including us, have used these little tiny worms to study longevity. So to look for genes that may be important in making the animal live healthier or longer. So for example, in the lab, we have a dietary restricted worm. So we can dietary restrict these worms, make them eat less, and then they become long-lived and healthy. So this is one of the longevity paradigm that it's a long-standing longevity paradigm that's in the aging field that all of us are aware of, that dietary restriction in the worms can lead to a longer life and also a healthier life for the worm. So in these long-lived worms, we also found that they have very efficient recycling process. So therefore, our approach is that now imagine if we have identified these beans and trucks that I talk about. Maybe I can make the beans and trucks in the normal worms become more efficient, and then maybe they can live healthier, and maybe comes with a bonus with longer life. Who knows? So that's where we are trying to get to. So the last questions that I want to leave with all of us to discuss is that, can we boost cellular recycling in humans? So far, we know that in worms, when we dietary restrict the worms, they can live longer. We also know that in yeast, humans, we don't know about the human cells yet, but yeast, fly, and mouse model, it has been proven that if you dietary restrict these animal models, they can live longer. And we don't know, what we know of is that while dietary restriction can extend the lifespan and these animals live longer, we also know that if we take away the recycling process, these animals will not be able to live as long as they did when they first dietary restrict. So we know that dietary restriction, as well as other longevity models that I didn't mention here, all require this recycling process. But we don't really know if there is a direct linkage between it, but we do know that it requires it. So in human, while we don't know the clear answer to it yet, which is a constant effort that the scientific community is trying to figure it out. But I hope you can appreciate that the effort that has been done so far, that we are coming from a really long way and we still have a really far way ahead of us, but that doesn't mean that it's not achievable. It's still in the progress. So with that, I would like to, of course, acknowledge all my lab peers who have given me uncountable supports and advice, especially my mentor, Dr. Milena Hansen, who helped guide me in my research career, and then all my fundings. And with that, I would like to take questions and we can open floor for discussion for sure.
Wonderful. Thank you so much, Ify. That was awesome.
You're welcome.
I like the analogy of the recycling in the real world. Also happens in our body. That makes it quite understandable, and I appreciate that. All right, audience, your turn. You have Ify sort of all to yourself for questions and discussions. So what would you like to ask Ify? And I'm going to stop her sharing just so we can see her better.
Oh, yeah,
there we go. This way we can all see her well. I think makes for better communication.
Mm-hmm.
And you might notice that I am recording this, but I'm not recording. I'm just recording the audio because we might want to turn what Ify said into a podcast. So I'm not video recording any video. So if you would like to start your video, you're welcome to do it, but you certainly don't have to. It just makes the conversation oftentimes a little bit more personal. So who's first with questions? You can either raise your hand or unmute yourself, or if you don't want to speak and you want me to read your question, you can put it in the chat. Either way is totally fine with me. Who wants to go first?
No questions.
It always just takes a second. And if nobody has, I can get us started. I have tons of questions, always.
Okay. But first of all, I would like to applaud you guys for, tuning in. So it says something about you guys, like you really care about the science that's going on in the scientific community. And hopefully we are here to make it easier for all of you to understand what we are trying to achieve in the lab. What is the action that's going on in the lab?
Perfect. We have our first question. Michael, do you want to go ahead?
Sure. We'll see if I can get it in before my 5-year-old. Nope, didn't happen.
Hi.
Here, you can ask a question with me. Here.
Mm-hmm. Hi.
How much of the, I listened to the Audible recently of David Sinclair's book on lifespan.
Mm-hmm.
And he goes into this a little bit, the brain chemistry and so forth. Mm-hmm. How much do you think, or how much is related to your research, do you think of this recycling of the mind, or could increase lifespan? I guess, like, how many years? How much of an improvement do you think of is potentially there through these processes?
I have no idea. So currently,
I wanted an exact answer.
you wanted a very specific, exact answer, which is it's gonna take a lot of time to figure that out. I think that's what we we don't know yet. We don't know the answer yet. However, I know that whether or not first of all, dietary restriction is it's kind of like a whole organism's things, right? When you reduce food intake, it's not only reduce food intake in the brain, but it's like the entire body, like all the cells in the different organs are also subjected to dietary restriction. So if you're talking about like, are you talking about like specifically dietary restricting the organs? In that case, I don't know. I don't know that yet. However, for the recycling process, we are still trying to, I can tell you what we're trying to figure out right now. So we are trying to figure out that if we can find a way to boost the recycling process in a tissue specific manner. So how is that going to affect the health of the entire organism? So maybe there is a particular tissue that in the animals that is preferring to recycle a certain type of cargoes. And then if we provide more of the bins and trucks or like all the equipments for the particular organs to do so, maybe it can improve the entire organism health and lifespan. In fact, we do have some like ongoing evidence is supporting that, but it's still an ongoing process. So I don't know if I directly answer your questions, but I'm pretty sure I didn't directly answer your questions, but hopefully I can let you know more about like what we are currently doing in the lab.
No, that's good. Thank you. And I want to mute for most of it because I do have the little 5-year-old. But you're talking more, I guess, 1 second, sweetheart. You're talking about two different but related things, which is health span, what you're calling, I guess, which is quality of life versus lifespan. Mm-hmm. So do you think your research in the process, but what you're researching, is that more likely to see improvements in health span, quality of life, or total lifespan? Which one do you think is going to be more the recipient of your work?
Yeah, so for me and our lab, particularly, we really focus on the health span more than the lifespan. I think the lifespan is a bonus. So you can have a really long lifespan, but if your quality of health is deteriorating, the remaining of the lifespan that you have extended, it doesn't mean anything. So that's what we are emphasizing on. So like to do that, most of the time when we do experiment, we might stress the worms. Let's say if we tweak the genes now, and then we saw that there's a lifespan effect, like now the worms are living longer. So the next question we will then ask is that, well, now if we stress the worm, can they overcome the stress? Can they be resistant towards the stress? So that's the indication that we will gather to figure out if this is really extending the health span while the lifespan is being extended.
Super, thank you so much.
You're welcome.
And we have some questions in the chat, so I'm just going to read them real quick. Mary is wondering, what is the recommendation for dietary restriction for humans? Do you have any idea? Do you have heard anything from your colleagues of what that should be?
I really don't know, and I am not a. I think this will be a good question for a dietitian. Yep. Yeah, I am nowhere an expert to provide any sort of measurements or recommendations. But in the lab, what I know is that we use a genetically manipulated worms, which it's no longer able to eat as normally. So that's how we reduce their food intake. So like it has been reduced food intake for the long time. But however, even though they have reduced food intake, but when other scientists have measured the concentration of the food that they have eaten, they found that it's a reduction of 20% of a normal worm. So at least in the worm perspective, like 20% of reduction of the dietary restrictions according to their nutrient scheme, can have this sort of lifespan or health span benefit. What you don't want to do is become malnutrition, so like not eat anything at all. That's actually detrimental, so you don't want to go there. So if you want to consider that, I would recommend that you talk to a dietitian. But in terms of research wise, so far all the dietary restriction benefits that has been shown is only in mice model, rats model, even like monkeys has been tested. Dietary restriction can extend lifespan, yeast as well as C. elegans, but not in human yet. So that is something that you should also consider.
Thank you. And we have a question here. Once you discover one of these recycling processes and a way to boost it, what is the process to apply it to humans, and how long do you think it would take?
So hopefully, once we identify this these different beans and trucks, so there could be a potential genes that we can target, right? There could be a gene. So now we can genetically introduce this genetically manipulate the animal models to make more of these genes. For example, we can use a CRISPR technique. I don't know if anyone have heard about it. It's a pretty hot technique right now that's in the scientific field. So you can tweak the gene, and then now the genes now the cells. So you can tweak the DNA in the gene in the cell. So now the cell would be like, okay, I need to make more of this gene now, which is eventually will be made into the enzymes or the proteins that I mentioned about in the earlier talk. And then hopefully this will become more efficient because now they have more of the resources to do the recycling efficiently. How long it's gonna take? I don't know. we will try. We will try to do it soon, but it's definitely not this year or next year or next year. So it's gonna be a while.
But there's hope inside.
Yeah, there's definitely hope inside.
Perfect.
Yeah.
And we have another question. Do you think it's possible to replenish depleted digestive enzymes?
Replenish depleted digestive enzymes.
Yes. So the digestive enzymes are gone.
Mm-hmm.
And is there a way to get those back?
Yeah. So I think it has to from what kind of digestive enzymes we were talking about, right? That's very important. So if we know of a specific digestive enzymes here, then we can find out in the cell what is making these enzymes. So then hopefully there could be already a drug that is available to can make the cells to kind of make more of the enzymes. Or if not, then we will have to find a way to genetically introduce that, but I don't know. I'm not really an expert in digestive enzymes, so I don't know what is the current trend or the ongoing studies or discovery that has been made in the digestive enzymes field. So, but I think it's like theoretically it's doable, but there's definitely a lot of steps that needs to be involved, and a lot of the cautious measurements needs to be taken to get there. But yeah, that's the answer that I could provide for that question.
That's perfect. And Peter would like to know, will your research become disease specific, such as Alzheimer's, colon cancer, and I can't even pronounce the last one. I'm sorry, Peter.
Atherosclerosis. Yeah, I am hoping that in the future, my research will become disease specific. So again, like I mentioned earlier, so one of my goals is to look for these cellular beans and trucks. So and that there's some growing interest in me that I didn't mention earlier, that I have been always interested in understanding the neurodegeneration disease development. So how does the neurodegeneration disease progress? So that's like my work when I was doing my graduate school. So hopefully with this knowledge that I gathered, I can now maybe use them, find out if whether or not these beans and trucks can be effective targets for slowing down Alzheimer's disease progression, which we can try to do them in a cell line obtained from patients that have Alzheimer's disease, and then just treat them with genetically manipulate the genes that or the proteins that I have talked about. If I identify them, make the cells make the Alzheimer's disease patients cells to have more of these beans and trucks, and then see how the cells now can recuperate or not, or like become healthier. So it can be more disease focused in the future. But what I'm doing right now, it's not there yet, because I'm trying to tackle the basic mechanism first before we translate it into a more complex model organisms.
That's perfect. And we have another question. Does the increase in diet restriction reduces the activity of the model organism and overall energy consumption of the organism over its lifespan?
Does the increase? Oh, that's a very scientific question.
Yeah.
So, no. So the dietary restriction actually did not. I don't see, at least in my hand, for the animals that I handle with the C. elegans, I don't see that they have reduced activities. They are happy. They can still swim fine and everything. Whether or not the energy consumption, I have never directly measured it, so I don't know. I don't know the answer to the questions. I haven't measured the energy consumption. So in the lab, we have a very specific way to measure energy consumption. So there are kits that we can use to do so. So I personally haven't done that yet, and I haven't read anything that's relating to that. So maybe in the future, when I encounter this audience again, I can provide him the answer once I figure that out.
That's perfect. Well, at least we give you some new research ideas, and I like
Yeah.
the back and forth between, the exchange between the speaker and the audience. That's pretty perfect.
Yeah.
We have another question. Is there any correlation to this research in combating COVID or any kind of virus infection? Does the autophagy have any influence on
so for my research scope at this point, I'm not doing anything related to COVID, but I do know that the autophagy recycling process is able to recycle bacteria, and also it has the ability to actually so the recycling process have like a vesicle. It's like kind of made out of a vesicle. So in the scientific way that I'm trying to explain it in a more easier ways, that we do know that while this recycling process normally can recycle things, it can also serve as a carrier to carry like some of the antigens, like to secrete things that could help fight against viruses. So I don't have any knowledge on that, but I do know that there are people out there in the autophagy field is trying to tackle the questions of how autophagy can be playing a role in helping to combat COVID.
Wow. And there's so much work around COVID that I
I know.
haven't heard about yet, which is amazing. I love that there is so much being done.
Yeah. All these processes that has maybe just not me who talk about it, but other processes, they all play very important roles in helping the cells to survive. So this particular process I'm talking about is just handling the recycling, but there are other processes that I probably are no expert in talking about. They also play an important role in making the cells come together and work properly.
Yeah. Speaking of trash, how do you look for the trash in the cells?
Very good questions. So in the cell, we can add fluorescent protein, so we can make the trash light up. So for example, I'm talking about mitochondria, right? So within the mitochondria, it is actually made up by a lot of the different kind of mitochondria proteins. They come together and then they made up the mitochondria, and then they do its function. So what I can do is that I can tag one of the mitochondria proteins with a fluorescent tag. So then I can genetically introduce this tag protein into the worms. So now the worms, all the mitochondria proteins that's being tagged with the fluorescent is going to light up. So you can see where the mitochondria is at. And then you can look under the fluorescent microscope and then track it in the cell. So this is just one example. You can do the same thing for the fat. So like the lipid droplets has some proteins that comes in between the fat droplets, and then you can tag it with a fluorescent, and then you can trace them. So normally these cargoes, when they should be degraded after a while. So but during aging, especially when we use the worm, we can see that some of the thing that we tag can start to accumulate. That could be associating with like a decline in the recycling being take place. So that's why you have the accumulation of the trash. But there are other ways that we could do so to make sure like we can, like all this damaged proteins that I talk about, like mitochondria, sometimes they can have a tag. You know, it's like let's say, imagine your car broke down at the side of the road, right? So the tow truck people will kind of put a sticker on, and then so now the tow truck will come by. The tow truck officer will come to the location and then put a tag on, yellow tag on. And then eventually on the freeway, you will see then now the tow truck came and then see, oh, there's a tag on this car, I'm going to tow it. So the trash has the same tag too. So if it is a track, if it is a trash, it can be tagged with a yellow tag in the cell, yellow tag. And then now we can follow this yellow tag together with the protein that's already be fluorescent tag. So you have a fluorescent protein that's tagged, and then now you have the yellow tag, and combined with that, then you know that, oh, this is a trash, and it's not a health. It's not because it's accumulating because it's a trash. It's not because it is not healthy. Well, this is just a very simple way that, and one example way that you can distinguish the trash and non-trash.
Perfect. So I know there's always a long ways for basic research. I know you call that basic research, although it's not basic in many ways. But, from going to basic research into human research, how long does that normally take? And when do you think from your worm, are you going to move into mice? Are you going to move into something else before you go to humans? Or what would that process look like if you ever go into humans?
Yeah, I think most of the time it depends on the researcher, like herself or himself. So for me, for example, I'm currently working on a project which I'm trying to make so the original findings was made in human cells. And then after that, I wanted to figure out how this process would take place in C. elegans, right? And then I did the experiment and found, oh, there's something interesting that's happening in the C. elegans. Let me go back to the cell line and see, oh, if this process is conserved. So it's interchangeable. But what is very complex and what takes the longest time is actually going to a more, like human cells is kind of like the simpler model from human. But the more complex ones is the like mice or like it's like the whole model organism, just because they have more organs and to tweak the genes takes longer. So like, for example, if I want to create a genetic model of the worm, like if I tweak the genetic model, it just takes me like a month, I can do it. But to do that in a mice, it's going to take years, because first you have to take the eggs of the mice, and then you have to introduce the genetic mutation, and then you have to mate them, and then you have to expand them, and then you have to verify if the gene is correct or not. So that process takes time and sometimes takes years. So that's the reason why it takes so long. So how long, I cannot give you an answer. It just depends on whether or not the resources is available to you to begin with. Maybe you are lucky, the thing that you work on has a genetic model already in the mice model already available, so you can translate it right away. But sometimes it takes years because you have to start from scratch and you have to do the steps that I just talked about. But in worms, it's pretty fast. In cell line, it's also pretty fast. What is really slow is the mice and the rat, and like to even have the confidence to go into human. That is what takes slow. The quickest way to do that is to do everything together. So like everything is going in parallel instead of like doing one step at a time. But usually that takes a lot of manpower. You have to have so many people comes together and work on the same project, and you tackle all the model organisms at the same times.
Yeah. And you look at the autophagy and the trash removal for different things, for neurodegeneration, for cancer, for heart disease. Where do you think in your research, where do you see the most promise right now? Do you see it in neurodegeneration, that you will make some progress and can help humans with a healthier, longer life?
So luckily for the worms, they don't really develop heart disease because they don't even have a heart, and they don't even have, rarely they have cancer. You don't even see them developing cancer, or they have any sort of maybe they have some neurodegeneration that you can observe from. For my current research, I am mostly interested in the fat recycling. So I think this research will mostly benefit maybe like cardiovascular diseases or even neurodegeneration, because now there are a lot of scientists has now discovered that the accumulation of fat has a very high correlation with neurodegeneration diseases. So maybe this will be the two spectrum, or like the area that my research will be most benefited. But then if you talk even more deeply, you can also say that all of them are kind of interconnected, because a lot of the studies now have found that obesity, which is associating with a lot of accumulation of fat, has people who are obese or like have a very high chance of getting high blood pressure or diabetes, and even with cancer. So in a way, they're kind of interconnected. It's just, it doesn't know who comes first, and how it happens. So I think my research, I think the very direct one will be like heart disease or like obesity, and then neurodegeneration, and then maybe eventually it will be benefiting for cancer and other diseases, but not right now, because I'm using worms, which I mentioned that it doesn't really have any heart disease because it doesn't have a heart. But the basic mechanism is similar. They share very similar recycling processes.
Perfect. So before I'm going to take a look at the chat again for questions, and before I otherwise ask my next question, I'm going to launch the poll. So if everybody, while listening to questions and answers, if everybody could take a second to take that poll, that would be much appreciated. Like I said, it gives us good information about how we're doing with the talk, and also some demographic information that we like to know about. It's always helpful to know.
Mm-hmm.
All right, so in the poll, we don't have a question right now, but I have a question, obviously, always. So you say that autophagy, the decrease in autophagy comes with age. Is there a particular age in humans where that starts, where it starts to decrease?
In human, I feel so bad because I cannot, I'm trying to do research that help human, but I don't really know most of the thing that happening in human yet, because it's very hard to just take human samples and and just do the research. But I know that in worms, that so worms live up to 21 years old. Let's say the lifespan is about 21 years old. I think like by day 7, so it's like their midlife time, they already start to show signs of declining in the autophagy process. So I would say maybe that, I don't know, but maybe it could be very similar trend for human too.
Yeah.
Yeah, but that's a very good question. And you say autophagy right too. I'm very proud of you. I think I did a great job.
Yeah, you're a good teacher. Perfect.
Thank you.
For me, there's always the ethical question, right? I mean, we're trying to prolong human life, which I think is there's an ethical conundrum with that, because there's already an issue with overpopulation and resource scarceness. How do scientists approach that? Like, do you in the lab discuss, should we do this research? What could be the possible consequences of it? How does the ethical world play into your decisions and your research?
So it's very interesting you ask this question, because I asked this question too when I first so a few years ago I went to a aging course, and then that's how I met my mentor, and then she got me interested in working in autophagy. And at that time I was so scared, you know, when the instructor was telling us, oh, now we can extend lifespan in mice model, and then in C. elegans model, in this model, and they can live so much longer. I was kind of thinking, what about the society? Can we compensate for now all the centenarians? If now we start to have, maybe in the future, we start to have a lot of the people now can live longer. Can we compensate for that? So I don't think now we are there yet. However, I kind of see this as a positive thing. I think the most important thing is that we have a lot of people who are healthy when they become older, rather than they live longer. So I think that's the goal that we are trying to focus on, is can we help human to have a longer health span rather than a longer life. That's what we're trying to tackle. So, and if we happen to have a lot of the human that has healthy life while they're living longer, that's actually very good for the society. That means that we can find ways to have more job opportunities, or maybe some people who are older right now, they can go back to school and learn new skills. So I know like in Japan, like Okinawa, they have a lot of centenarians, and they already start to implement like some sort of kind of resources to kind of help these centenarians to keep going, but at the same time not causing too much of a burden in the society. But you also need to look into a second factor, right? It seems like the reproduction rate, like in the society, is kind of like decreasing. A lot of people are starting to have bear their child at an older age. So I don't think we're going to be overpopulated with a lot of maybe I'm wrong, yeah. But I'm just after a while, I just look at all the talks, and then I talk to all the people. I just don't think this will be a potential problem. I think the problem now is that like with the declining in the reproduction rate, like especially like the younger generation now, they kind of usually put reproduction on hold first, and then they go for their career, and then later. So maybe this may be a good thing if we can find ways to extend health span rather than focus so much on the lifespan. And this will definitely help reduce the burden of the society to need to take care of a lot of when we get old, and then we become sick. And then now we are relying on all the healthcare resources that could maybe be utilized for helping other people, you know. Yeah. So yeah. That's my perspective, but may not be the most perfect perspective.
Well, but I'm just glad to see that you guys discuss it, because I think it's important for the ethics of of
Yeah.
research.
Yeah, in our aging course, we discuss it, and my instructor also talked to me regarding about that. I don't think I think the main goal right now, especially in the aging field, is we are not really emphasizing on living longer, but we really emphasize on living healthier and how to extend the health span.
Yeah, that makes sense. And I see that Mark and Anne have a question, so I'm going to ask them to unmute so they can ask.
Mm-hmm.
First of all, we don't seem to have the raise hand button this week.
Oh, well,
I think it's in the file. Oh, sorry.
I couldn't find it, so I
Okay.
just typed it. So how much do genetics play into this? I'm assuming that your worms are kind of like, the white mice that are used in a lot of research that are, genetically almost identical from copy to copy. Humans, are much more genetically diverse. So how much do genetics play a role in the waste transport system? I mean, I don't know whether the worms are, you're essentially studying the same worm over and over again to control for variability on the genetics. I'm just wondering, how genetics play a role in this.
Yeah, genetic definitely plays an important role in this. And I can tell you that while it's true that in human cells, there are a lot more diversities than the genetic in the C. elegans, but a lot of the core components of the recycling process is in fact pretty conserved. We have a few proteins or like genes that only exist in the human and we cannot find it in the worms, but a lot of them we were able to find them. Of course, one of our goal is that once we find them, then we will test if they do the same thing like their human cell. So the percentage, are you asking for a specific percentage of like similarities between genetics of human compared to C. elegans?
No, I'm more looking for, how genetic variabilities within a population. So within a human population, one person to another, within a worm population, between one worm and another, affect the efficiency of the recycling system as you've described it. In other words, I'm assuming that most of the worms that you're studying are genetically identical or similar. What if you had two worms of, a fairly different genetic makeup? Because, humans have, one overweight human might never develop diabetes, but another overweight human with the almost identical diet will be very prone to diabetes. So there seems to be a genetic component in some of this. And I'm wondering how that plays into your studies.
I see. So for me, when I do my research, I just get a lot of worms. So I don't think I take into account or like measure each worm specifically their genetic backgrounds. So I would say it's a very good representation of what is going on in the society. Because like in the worm, it's true, they also have genetic variabilities like human, but they might have a smaller genetic variabilities compared to the human pool, just because we control for the experiment, right? We feed them with the same food. They probably go through the same lifestyle like other worms. So that is something that we control of. But whether or not how the worms respond to genetically to the environment that we provide to them, we have wild type worms that we know that they are from the wild and they don't have any genetic manipulation that we have introduced artificially. So we have the wild type group that very similar to, I would see them as very similar to human, just maybe not as great as a genetic variabilities compared to human. But there are some worms that we will genetically manipulate them. but they're also coming from different background too. So in this case, I feel that it's pretty average. But specifically each worm that we are study, we don't study worm by worm, but we study group of worms together. So hopefully that would take into account like for the variabilities that's taking place in real life. But obviously they are not really the same as human, because humans are very complex, but they could be very powerful model organisms to just figure out the basic mechanism first, and then use that to go into human cells and see if it works the same. And if not, then we can find out what is not working the same. Why? Maybe it is less complex. That's why certain parts is not needed, you know, or certain parts can compensate for each other because they are less complicated. Did I answer your questions, Mark?
Within the scope of this meeting, yes.
Perfect. Good answer, Mark. Perfect. But I think that was also a great question that to close out the talk, because we've just come up to 8 o'clock.
Yay!
Yay! You made it, Evie.
I made it.
You did really good.
It felt like my committee meeting when I was getting my PhD.
I can share with the audience that Evie was a little bit nervous because it was the first time she's done a talk like this, where it's more conversational and more questions than your regular lecture. So Evie was a little bit nervous, but I think she did exceptionally well.
Thank you. Yeah,
Thank you so much.
Yeah, and if you have any questions, if I didn't answer, like get to answer, or like you feel that it's not sufficient, feel free to let Andrea know, and then we can extend the conversation through emails or later on. Yeah, exactly. Or I can consult my mentor, which is she just showed up on the screen, Melina, with the sunset background.
We're also quite honored to have Dr. Melina Hansen on the call. She joined us tonight. As Evie said, she's the mentor and she runs the lab at SVP for this research. So, hi, Melina.
Thank you.
All right, so that concludes the talk tonight. I just want to remind everybody that we have other talks coming up. We have, on October 12th, we've got all the small things, how tiny organisms make a huge difference. And we're going to look a little bit at dirt, basically. And we see how fires and things can have an effect on our ecosystem. On November 9th, we're going to look at deep sea marine biology. Excuse me. There's some music going on in the background. And we have our Sharp Minds lecture. If you're interested in more of a straight-on lecture with 45 minutes of PowerPoint and then about 15 minutes of Q&A, we have our Sharp Minds lecture that happens on the first Monday of each month in the evening. The next one is going to be about crime scene reconstruction, and after that we're going to talk to a biological anthropologist, big words for me to pronounce, and she's going to tell us a little bit about her research on bones and remains from the turn of the century and what those can tell us about the life at the turn of the century. So all exciting stuff coming up. Thank you so much for joining us for tonight, for listening to Dr. Evie Tan, who was just fabulous. It was great. Thank you so much, Evie.
Thank you so much for the opportunity.
Yeah, glad.
And thank you, Melina, for coming.
Perfect. Wonderful. With that, thank you all so much, and I will see you hopefully again next month.
Okay, thank you. Bye-bye.
Thank you. Bye, Evie.
Bye.